This is the second set of lightning talks. We have 5 speakers here who have posters up as well. We'll kick it off. Same rules, 8 minutes, 7 minute. I'll give a 1 minute warning and 30 seconds in. The first presenter is Lu Li from Penn Medicine. She's going to be talking about synthesizing evidence for rare events.
Hello, everyone. My name is Lu Li, a PhD student at the University of Pennsylvania, advised by Dr. Yong Chen. Today, I'll be talking about synthesizing evidence for rare events, a novel zero-inflated bivariate model to integrate studies with double zero outcomes. Let's start with a real-world case study taken from the Cochrane database of systematic reviews. The authors explored the potential use of probiotics as a treatment for Clostridium difficile-associated diarrhea, also known as CDAD, caused by antibiotic use. And specifically, they want to know whether probiotics cause any side effects when used to treat CDAD. You can imagine that this meta-analysis was in fact an OHDSI network study where each data partner contributed some estimates that are being pulled together. And as you might know, adverse events are often very rare. In fact, among the 32 studies in their meta-analysis, 10 of them have no events in both the treatment and control groups. Such studies are called double zero event study, or DZS. Double zero event studies often arise in OHDSI research. In fact, the larger the OHDSI network study is, the more smaller sites will participate, and the more likely it is that we will see these double zero scenarios occur. For example, the Darwin EU, as we've seen in the previous presentations, is a federated data network consisting of many healthcare databases across the European Union. And they typically have smaller cohort sizes, making them especially likely to encounter double zero studies. The problem is, double zero studies create challenges for estimating commonly used effect size measures, such as relative risk and odds ratios, due to the division by zero error. So the question then is, how should we deal with the studies that had no events? A natural approach might be to exclude them from the analysis, and is in fact commonly done in practice. However, there have been studies showing that the exclusion of double zero studies impacted the conclusions. And furthermore, since we want network studies where anyone can contribute, we need a solution to handle the double zero studies. One approach to include the double zero studies in the analysis is to use some one-stage methods, for example, the bivariate generalized linear mix models, or BGLMM. The BGLMM is a bivariate random effects model that jointly analyzes the risk in the treatment and control groups. And specifically, the number of events for the treatment and control groups are denoted as Y and follow a binomial distribution, with N being the sample size and P is the event risk. And event risk are modeled using a fixed effects and random effects component, and the parameters of interest are the fixed effects and random effects, and they can be directly estimated using the maximum likelihood estimation. However, one limitation with the bivariate generalized linear mix model is that it treats all the double zero studies similarly to the other studies. The question then is, should we treat the double zero studies similarly to the other studies? The short answer is no, and let me explain why. The rationale here is that the sample size of the studies are informative. And let me explain this intuition through a simple example. Assuming an event rate of 1%, the chances of observing a cohort with 110 double zeros is 1/3, which is probable. Whereas the chances of observing a cohort with 1,000 double zeros is around 4 out of 100,000, which is very unlikely. And therefore, the actual event rate for the large double zero studies might be much lower than 1%, indicating the fact that they might be coming from the extremely low-risk population. So our conclusion is that we should not treat all the double zero studies as the same as the other studies. So to differentiate the large double zero studies, which might come from the extremely low-risk population, we used the zero-inflated models, which separate the observed zeros into two distinct categories. The first category of zeros are known as the at-risk or chance zeros, which correspond to the latent group of population who are at risk for an event but have a recorded count of zeros by chance. And the second group of zeros are known as the structural zeros, which represent individuals who are not susceptible to a specific event and thereby having no chance of a positive event count. And here is the formulation of a zero-inflated binomial model, and the event number follows a binomial distribution with probability 1 minus pi, or it is definitively 0 with probability pi. So our method combines the zero-inflated models with the BGLMM, and is called Zero-Inflated Bivariate Generalized Linear Mixed Models, or ZIBGLMM for short. So our model is a two-component finite mixture model, and given a double zero study with zero event counts for both treatment groups, it either comes from the at-risk population or it comes from the low-risk population. And still, the parameters of interest are the random and fixed effects and can be estimated using the maximum likelihood estimation. The advantages of our method are twofold. First, no studies are dropped from the analysis. And second, our method models the population heterogeneity, by which I mean the existence of a low-risk and an at-risk population, through a data-driven approach. Now let's revisit the case study. Recall that 10 of the 32 studies are double zero event studies with sample size ranging from 18 to 144. In their original analysis, the authors excluded those 10 double zero studies and concluded that probiotics reduced the risk of adverse events by 17% with an RR of 0.83. Using our method, we included the double zero studies, and the RR we obtained is 0.70. So our conclusion is that including the double zero studies could potentially lead to estimates that differ by a large degree, i.e. greater than 0.1. So in summary, zeros in the double zero event studies may arise due to population heterogeneity. And second, ZIBGLM offers a more comprehensive analysis of the available data. For OHDSI, ZIBGLMM is especially useful for larger network studies and for studies involving rare events. If you have any questions, please feel free to stop by my poster number 506. Thank you for your attention.
Thank you, Liu. Next up, we have Kevin, who's going to be talking about the active safety surveillance using the real-world evidence, the Strategus package.
Great, thank you. We wish to thank the scientific committee for selecting this abstract, and I assure you that there will be audience participation required in this talk. I am presenting on behalf of the Johnson & Johnson Active Safety Surveillance Using Real-World Evidence team, or the ASSURE team. We are a multidisciplinary team with the support of our Global Epidemiology Organization as well as our Global Medical Safety Organization. We are tasked with rapid delivery of high-quality evidence to support active medical product safety surveillance. We utilize, I think everybody has seen this slide, we utilize real-world data to standardized in the OMOP CDM across multiple databases, and the OHDSI standardized analytic tools within the Strategus framework, thank you to the development by Anthony Sennen, which was presented at the last plenary. Strategus, as you know, is a set of R packages for coordination and execution of analytics within the Haynes modules. This standardized design takes the following inputs: target, or our drugs of exposure of interest, comparator, indication, outcome, and the time at risk. This analysis package then produces the reliable evidence that we need to make the decisions within our organization. We get a shiny dashboard to document the cohort diagnostics, the cohort characterization, the patient level prediction, and as you already saw with regards to the population level estimations from comparative cohort and self-controlled case series. To date, we have responded to over 20 requests, we just got another one last night, and delivered results within an average approximately about 45 days. Some requests close very quickly due to not being feasible, which is empirically evaluated through our database diagnostics. Some are also not feasible based on the inability to translate the outcomes into computable phenotypes. Other larger requests often take a few days of compute time or phenotype development activities that are ongoing within our team. Assure fits into the life cycle of signal evaluation portion of the life cycle management of safety signals within J&J. We work collaboratively with our medical safety officers and therapeutic area epidemiologists to provide timely evidence for final decisions and discussions at safety management team meetings. To date, this work has provided greater support to our established products group, but we have actually crossed all therapeutic areas with requests. A typical discussion often starts with a medical safety officer and is focused on a marketed product and an identified safety concern from our pharmacovigilance activities. Most of our time is spent on characterizing the clinical concept and translating that into a workable computable phenotype if one does not already exist within our library. An example of these are, you can think of those two Save Our Sisyphus challenges as the types of examples of the research questions that would come into our organization. This is the standardized set of packages. I just want to spend a few moments on this. These analyses implement the methods that Martijn Schumi and Patrick Ryan discussed at this symposium last year and throughout the year at our Save Our Sisyphus Challenge. Briefly, we utilize the database diagnostic tool developed by Claire Blackadder to identify candidate data resources for analysis across our internal network of available resources. We utilize the comparator selection tool developed by Justin Bond, Dave Kern, and others to identify the candidate comparators so that we're utilizing empiric evidence to select those comparators. We utilize our phenotype library that's internally managed by AZA to select our T, C, I, and O cohorts. We select negative controls for the empiric calibration with the methods outlined by Erica Voss at the 2019 symposium and subsequent presentations. And our JSON specifications that get created out of this that you saw that Senna was presenting at the last plenary are translated into a highly documented protocol with a tool developed by Jenna Reps for sort of ease of review and translation of the JSON specifications into a human readable document. These results are viewed in an interactive Shiny application with development from Nate Hall, Jamie Gilbert, and Jenna Reps and others here today. And we're very specific, as you saw in the Save Our Sisyphus Challenge, we're very specific that we only unblind results which have passed study diagnostics, outlined at last year's plenary and discussed at subsequent symposiums throughout the year. Where possible, we synthesize evidence across databases, and even when analyses fail, we have robust population-level characterizations of our target, comparator, and outcome populations to support the signal evaluation activities. This is a typical day, but our team meets twice a week and bi-weekly with our leadership, with Josh Gagne, Courtney Davis, and Patrick Ryan, all of which receives excellent project management support from Ella Blue and earlier this year, Emily Yost. We have additional meetings with our global medical safety officers and our therapeutic area colleagues to clarify those requests. And so a typical day may have us doing any one or all of these tasks as we work to develop and identify the needed phenotypes. We may have to work with a disease advisory council, which is a cross-functional group led by Gautam Rao. And Mitch Conover and I have crafted a lot of standardized templates, so we're getting there, Mark, with regards to some presentations and standardized reporting so that we can deliver this evidence, both to the actionable individuals that will take action on our questions and answers, but also for our reporting. So here's the interactive portion. So this is going to require some level of effort. This is my talent, right? Give me a T. T. Give me a C. Give me an I. Give me an O. What's that spell? Strategus. I know, you all thought I was going to say TCIO. Thank you, that's my only talent. Please stick around for the talent show. That's it. This is a snippet of code that from the Strategus tool, this creates the JSON specifications, and as you can see, it takes the inputs of a target ID, comparator ID. This looks simple because you're only editing above this line. As Martijn has told us multiple times, you probably don't want to change below this line. But there's a lot of work that goes into each of these requests to clarify each of these Atlas cohorts as we build this out. And we specify the negative control concept set, we specify our times at risk, and all of this is developed very collaboratively. So to learn more, please see one of our team members or stop by poster 507. I really thought that the Oscar music was going to be playing by now, but I guess I speak fast. So thank you, Justin Bond, Jenna Reps, Gautam Rao, Mitch Conover, for a wonderful year. It has been an absolute journey as we've implemented this across our organization. Obviously, the entire OHDA team, the Odyssey community. I'd like to thank my manager, my parents. This is where, all right, thank you.
Thanks, Kevin. Next up, we have Jessica, who's going to be talking about patient outcomes after ERCP.
Hello. So, hello everyone. I'm very happy to be here today. My name is Jessica, and I work as a real data service head in a startup from Brazil, which is called Precision Data. Today, I'm going to present this project that we conducted in collaboration with our partner, Boston Scientific, and is entitled Patients' Outcomes After Endoscopic Retrograde Cholangiopan Creatography, which we refer as ERCP from now on, using reprocessed duodenoscope accessories. This is a descriptive study using real-world data. So the ERCP has significant impact on the management of prognosis of pancreatic diseases. However, there are serious concerns related to the duodenoscope-related infections due to this material reprocessing. So this study aimed to compare the percentage of readmissions post-ERCP procedures between single-use and non-single-use institutions using an OMOP standardized data set from Brazil. As the data sources, we included the Hospital Ambulatory Information System from Brazilian Administrative Data Sets, which is called DataSUS. And these data sets were mapped into the OMOP Como data model. And to connect these both data sets and create the patient journey, we developed a deterministic linkage algorithm to connect this data set. If you want more information details about this algorithm, please visit the poster number 16, in which we give more details on how this linkage was done. We included patients from January 2020 to January 2023, and the inclusion and exclusion criteria were we included patients with no history of cancer, and also patients that had an event of ERCP procedure, excluding those cases that had this ERCP procedure due to sepsis, acute pancreatitis, or cholangitis. And our outcome of interest was the hospital readmissions up to 30 days after the ERCP procedure, and the causes for readmissions included sepsis, acute pancreatitis, and cholangitis. So the identification of the ERCP procedures was following a specific data source code system, and the statistical analysis was done in Atlas. The identifications of the single-use and non-single-use hospitals were done by our partner, the Boston Scientific, through field work. So they identified the institutions that by comparing the number of devices that they sold to these institutions with the number of procedures that they done. So if the number of procedures were higher, then they concluded that it indicates that these institutions were reprocessing the materials. So here we have some results, some characteristics of the 4 cohorts that we included in our analysis. And in comparison to the readmitted patients from single-use institutions, the readmitted patients from non-single-use had a higher proportion of female individuals and patients with a lower mean age. So in here, the difference between the non-single-use group and the single-use group, we can see that the non-single-use group had a higher percentage of readmissions of approximately 65% compared to the single-use group. So as a conclusion and next steps, as far as we are aware, this is one of the first studies using real-world data from Brazilian administered data sets and map it into the OMOP model. And this is especially focusing on devices, medical devices, and patient outcomes. So this is very important for us because we would like to enhance the use of this type of methodology into more institutions in Brazil. So we found a higher percentage of readmissions in non-single-use institutions compared to single-use institutions. However, we do acknowledge that this is just a descriptive analysis and that the characteristics between the patients and the institutions may not be the same. So we must conduct an estimation study adjusting for potential confounders and also for the unbalanced data. So this is our next steps for the project. And we really believe that this type of data also is very important to inform on clinical decision-making, optimal ERCP management practices, and leading and focusing on patient safety, which is, I guess, the purpose of OHDSI community. So thank you very much, and please, if you have any questions, we will be in the poster. I would love to hear your thoughts and ideas about this project. Thank you very much.
Thank you, Jessica. Everyone's keeping great time, which is really odd for an academic conference because no one's going over. So fantastic work. Next up, we have Bingyu, who's going to be talking about COVID increasing racial ethnic differences and prevalence of PASC and long COVID in kids and adolescents. After you, Bingyu.
Thank you for the introduction. Hi everyone, I'm Bingyu, a third-year PhD student at the University of Pennsylvania, advised by Dr. Yongchen. So today I'm going to talk about whether COVID-19 increases the racial and ethnic differences in the prevalence of PASC along COVID in children and adolescents. This is the work using an EHR-based cohort from the Cohort program. So let's start by understanding what PASC is. When people test positive for COVID-19, they are in acute infection phase, which lasts for 4 weeks. Some people may have symptoms while others do not. However, for many, this experience does not conclude after 4 weeks, where symptoms may persist or new symptoms may emerge. They move into the post-acute phase. So this extended experience is termed the post-acute sequelae of SARS-CoV-2 infection, also known as PASC. PASC is more common than you think. The CDC reported that among adults with COVID-19 who did not require a hospital stay, 2 out of 3 had at least one outpatient visit 1 to 6 months after diagnosis. Also, from one of the systematic reviews of long COVID in children and adolescents, the prevalence of PASC symptoms and conditions is around 25%. Although children and adolescents have fewer instances compared to the adults in long COVID, they can pose greater disease burdens as their parents have to provide ongoing care for them. So here, RECOVER is initialized by the National Institutes of Health to learn about the long-term effects of COVID and to predict, treat, and prevent PASC. RECOVER has PIs for adults and pediatrics separately, and our group oversees the statistical analysis for the RECOVER pediatric cohort. Here are some selected publications on PASC within RECOVER, such as burden, vaccination, and cardiac complications. Another concern is the racial and ethnic differences in the prevalence of PASC. CDC indicated that several studies reveal that, certain racial and ethnic groups and communities are more likely to go to the hospital related to COVID-19, which is due to social factors. For example, the Black or African American community is 2.1 times more likely to go to the hospital compared to the white group, while the Asian community is 0.7 times compared to the same white group. Moreover, a paper published on the Journal of General Internal Medicine found that compared to the white group, the Black and Hispanic patients have a statistically significant different odds of developing PASC symptoms and conditions. So, here comes our clinical question. Does there exist racial and ethnic differences in potential PASC symptoms and conditions among children and adolescents after SARS-CoV-2 infection? A classical solution is the standard regression model adjusted for measured confounders. Also in 2020, this legend paper suggested first fitting a large-scale propensity score model, followed by stratification, matching or weighting, and then running the outcome regression model with the propensity score adjusted. However, in our setting, how many of these differences are attributable to COVID infection? To account for the differences that are attributable to COVID infection, the difference in differences approach offers a robust solution. Here on the x-axis, we have the number of days since infection within an individual, and the y-axis is the prevalence of potential PASC symptoms and conditions across the population. Earlier research primarily focused on the purple rectangle here, that is the differences after COVID infection. However, it's crucial not to ignore the differences that already existed before COVID infection. So here, the difference in differences approach works as follows. We divided the differences in two parts. The first represents the differences that already existed before infection, and the second is the differences that totally resulting from the COVID infection. So it's essential to note that we are assuming that the pre-existing differences remain consistent before and after COVID infection. And our primary interest lies in the yellow rectangle, that is the differences totally resulting from COVID infection. So in terms of our solution, we incorporated the difference in differences analysis in the outcome regression model so that we can control the pre-COVID racial and ethnic differences. So our retrospective cohort study is part of the Recover network. After applying the specified inclusion criteria, we included over 200,000 patients across 13 institutions in the US. Roughly half of them are non-Hispanic white, 26% are Hispanic, 20% are non-Hispanic Black, and 5% are Asian American or Pacific Islander. We excluded patients from other racial and ethnic groups due to limited sample size. Let's look at our findings. We included a comprehensive list of potential PASC symptoms and conditions, and we stratified the patients based on the severity during their COVID-19 acute phase. So here, when we speak of the minority groups, we refer to the AAPI, Hispanic, and the non-Hispanic Black, each compared to the non-Hispanic white group. Here, we use the risk difference as our comparative measure, and the blue boxes denote that at least of one of these comparative groups have a significantly difference of risk difference. For example, among the patients within the severe group, the Hispanic patients showed a significantly higher increase in the respiratory signs and symptoms compared to the white group, while the non-Hispanic Black group showed a statistically significant lower increase in the skin symptoms compared to the white group. In conclusion, the racial and ethnic differences related to COVID-19 vary across the racial and ethnic groups, the severity of acute COVID-19, and the particular PASC symptom and conditions. In summary, our study helped to understand the racial and ethnic differences in PASC after SARS-CoV-2 infection among children and adolescents. Our study covered a broad spectrum of the US pediatric population, which follows the legend principle 1 and 9 to generate evidence at a large scale and across a network of multiple databases. Our study handles measure confounders using propensity score matching and controls the pre-COVID racial and ethnic differences using difference in differences analysis. This aligns with the legend principle 5 to generate evidence using best practice and aim to minimize bias. As for future work, we will explore methods to adjust for systematic bias. Thank you so much for your attention, and welcome to my poster number 509. Thank you.
Thank you, Bingyu. And last but not least, we have Michelle, who's going to be talking about some of the exciting work from the eye care and vision work group.
Hi, everyone. I'm Michelle Rybar, and thank you for the opportunity to speak to you today on behalf of our entire eye care and vision research work group to give our first year update. So just like anything in Odyssey, our first year has been a journey, and I'll talk to you about how we got started, our initial steps, some milestones we've achieved, and now looking forward to our journey ahead and the next steps. So we got started in the spring of 2022 because it was a need was brought up by the American Academy of Ophthalmology, AAO work group on data standards. And this was mostly coming out of the desire for eye exam data to be included in data generation projects like All of Us, N3C, and Bridge to AI. And we quickly also discovered that the source terminologies hadn't been updated in a while, so there was missing concepts there as well. But our overall goals were to create access to large, diverse data sets of ophthalmic and systemic data to enable research in both and also at the intersection. So some of our initial steps where we identified tasks and then created subgroups for those tasks, and also subgroups that were aligned with different subspecialties of ophthalmology and optometry. We recruited colleagues to participate, and the National Eye Institute, along with the National Institutes of Health, created a data scholar position to help with this project. That's me, and that has been one of our main supports and ways of getting this done this year. So the milestones we've achieved so far is that as of a week ago, we had 122 registered members on teams, and we'd say roughly 40 of them are active, attending work group meetings pretty regularly. And of those, we had 13 trainees and 10 interns from the Bridge to AI Ready project. We're a multidisciplinary team, mostly consisted of clinician scientists, ophthalmologists, and optometrists, but some informaticists like myself and some other vision scientists. We've had meetings at least once a month, sometimes twice a month for the whole work group, and then meetings for each of the subgroups. So we've met quite a bit this year. And I can tell you I've had countless ad hoc meetings. I couldn't even, probably at least once a day. We quickly found out that we couldn't do this on our own, so we're really grateful for the Odyssey community for being there for us. And so we've regularly collaborated with many of them, but we've also collaborated with external organizations to make sure that our efforts are aligned with the needs of the community, but also so that people are aware of what we're doing and to drum up kind of support and interest. Also to help us get some of those concepts addressed. So several groups within our work group started looking at mapping those elements. And so in total, we've looked at over 3,700 ophthalmic data elements, and we've looked at 2 EHRs, EPIC and Cerner, as well as looking at different ophthalmology textbooks for source concepts. We're in the process of getting concepts added to both SNOMED and LOINC. And this is just an example of one of the graphs from our EPIC EHR concept matching paper. Poster 8 has information about both EPIC and Cerner, but this graph is pretty similar for Cerner as well. And you can see that roughly only 25% of the concepts had an exact match in an underlying source terminology. And the majority were either missing or did not have a specific enough concept. And in ophthalmology, one of the most common things that was missing was laterality, which is really important for your eye because you really need to know which eye has the disease. But there are also other specific modifiers for concepts that are really important in terms of how measurements were made or different methodologies that were missing. So we've really tried to get the word out that we're doing this great work. So we've been publishing papers, both that have summarized our work, but also kind of advocated for better standardization. We have created some iWiki pages. That's a wiki that's maintained by the American Academy of Ophthalmology, and that's sort of educational for that community in terms of what standards are and the work that we're doing. We've done a lot of different presentations at conferences and ophthalmology departments, posters, including a couple here, really trying to talk about the work that we're doing. And we've done almost all of it entirely through volunteer effort. So the support pretty much solely has been from the NIH with the data scholar position. And we're so far have 2 grant submissions, but haven't heard back on them yet, but we're hoping to have many more this year. We were really thrilled that Cindy Kai, one of our workgroup members, had her project selected for the SOS Challenge this year. And she's already talked about that, so I won't necessarily go into the details of it. But I will say the thing that has been really exciting about it is that as Cindy has and myself have presented this work to the larger eye care and vision research community, they have been really excited and really see the value of this approach. So we hope that we can have, maintain this momentum and have more network studies this year. So the next steps as we're looking ahead is we really want to pilot some of these new data elements that we're working on mapping. We have a set of about 5 or 6 sites right now that are in the process of getting those data into their OMOP data mart. And we're also looking at integrating ophthalmic imaging with our data marts. So we're getting started on this, the first steps towards that process. This year, we're really focused on more of the community outreach to make sure that people know what we're doing and to get more people interested and involved. And as I mentioned before, we're hoping for more network studies and for more funding support. So like any journey, we've had some roadblocks or traffic cones, and we've had some challenges. Probably the biggest one is just finding a time to meet. With busy clinician scientists across multiple time zones, we often can't all meet at once. We also have had trouble with these concept modifiers and sort of adding things to the source terminologies. So for example, we just submitted over 200 concepts to represent very simple visual acuity concepts within ophthalmology. And the problem is there's so many modifiers. You can imagine laterality, distance, whether or not you're wearing glasses, and so it quickly expands. So we're working with Loink to figure out a way to manage that. Also, as I mentioned, we've had limited resources this year, but we're hoping to change that. And we're also hoping to expand sort of our membership beyond just academic medical centers. So in summary, we've had a very productive year. It's been great fun working with everyone, and they're all over there, so they're great too. And we're working really towards our goal. I think we're going to get there, but we still have a lot more work to do. So please join us if you're interested. Our work group meets on Sunday from 1 to 5 p.m. And you can also see more information at our poster. And I'd like to thank a lot of the people here, and I know I've probably left people out, but a lot of people from the OHDSI community, the entire work group, and especially our co-leads, Carrie Getz and Sally Baxter, and the National Institutes of Health to help support this through their Data Scholar program. And if you want to find out more details of what we've been up to this year, you can come by our poster number 510. Thank you.
Thank you all. Again, a round of support to all our speakers today. That was it. Thank you everyone for the lightning talk. I think we're going to head back out for the poster session. There are going to be some folks who are going to be leading the poster works. We have Melanie doing data science. Where's Melanie? Can you raise your flag? Oh, she's outside. Okay. Andrew, you're going to be leading the methods research. Is he outside too? Andrew? Everyone's outside. Open source. Sikak? Nope. Really? Okay. Clinical applications, honey. Someone, yes, finally. Okay. That's going to go on till 5, and then we come back on and sit down. Yes.